Welcome to my presentation about standardization of concentration measurements of platelet-derived extracellular vesicles for medical diagnosis. I have one conflict of interest to declare. I'm a major stockholder of the company Exometry. I will start with motivating why standardization of concentration measurements of platelet-derived extracellular vesicles is important, thereby discussing calibration, reference materials, quantifying sensitivity by photon counting, and reporting. So please note that I will use the term extracellular vesicles, or EVs, for what has been called microparticles within the ISTH community. If we succeed in standardization, we can establish the first reference values. And I would like to finish by announcing relevant activities from the EV flow cytometry working group. Now, my goal is to develop an EV-based liquid biopsy, which involves drawing blood, preparing plasma, determining the concentration of EVs of certain cell types, and compare those to results of hematology reference tables in order to provide physicians with new clinical information. Now, to determine the concentration of EVs, flow cytometry has high potential, especially because flow cytometers can characterize 100,000 of EVs per minute. As you can see here, this characterization is based on measuring light scattering and fluorescent signals of single EVs illuminated by one or more laser beams. However, measuring EV concentrations is not trivial, especially due to their broad size distribution. Here you can see the concentration of EVs on a vertical log scale versus diameter. It is clear that the concentration of EVs decreases rapidly with increasing diameter. You can imagine that when you have two flow cytometers with a different limit of detection, for example, 180 nanometer to 250 nanometers, that you measure up to a twofold difference in concentration. Thus, to allow data comparison, EV concentrations should be measured within the same size range. Therefore, it is bad practice to report we found 200 million EVs per milliliter. Rather, report we found 200 million EVs per milliliter, ranging 180 nanometer to 600 nanometer in diameter. Now, another problem is that flow cytometers present data in arbitrary units. Here you can see a side versus forward scatter plot of submicrometer particles in human blood plasma. Because the data have arbitrary units, it is difficult to interpret the data. From this plot, it is even possible to confirm whether the detected particles are impossible to confirm whether the detected particles are within the EV size range. So to improve data interpretation and comparison, flow cytometers require calibration. In a nutshell, calibration means to relate arbitrary units to the standardized units. For example, after calibration, the flow rate should be expressed as microliters per minute. Scatters should be related to particle diameters in nanometer, and fluorescence should be expressed as molecules of equivalent soluble fluorophores. Without calibration, measurements result can be utterly misleading, as I will explain in the next 5 slides. Here, you can see the scatter signals of EVs in arbitrary units, measured with a BD LSR and BD influx. Although it is impossible to read the size of the measured EVs from these graphs, you may think that both flow cytometers measure the same size of EVs, as the bars have equal amplitudes. To find out, we measured the scatter signals of 200 nanometer and 400 nanometer polystyrene beads as a reference. Based on the outcome, it now seems that both flow cytometers measured about 300 nanometer EVs. However, light scatter signals are complex and depend on collection angles and particle refractive index. By applying Mie theory, we can take all these optical details into account and calibrate the flow cytometer, thereby revealing that the BD LSR measured 1160 nanometer EVs, whereas the BD influx measured 500 nanometer EVs. Thus, without the application of Mie theory, the measurement of polystyrene bead can be misleading and is not a proper calibration. Daily runs of polystyrene bead mixes, however, are still common practice in the field, as reflected by these 4 articles and these 356 articles more. Therefore, I think it's time for change. I recommend to turn your flow cytometer into a particle size analyzer. As explained by Leonie de Rond et al., this is easy. You don't need knowledge of me theory. It's cheaper than daily quality controls with polystyrene beads, and it's more accurate than nanoparticle tracking analysis, although that depends on the flow cytometer. The calibration of flow cytometers requires reference materials that are well characterized and have physical properties overlapping with EVs. At present, however, such reference materials do not exist. Therefore, we initiated the European Metrology Program METFAS2 to develop one bead to calibrate all flow cytometry aspects: number, concentration, light scattering, and fluorescence using reference particles that resemble EVs in their physical properties. As an example, the transmission electromicrograph on the right shows hollow silica beads made by Zoltan Farha. And if you want to find out more about their applications, please visit medfas.eu. Now, even after calibration, when having access to the diameter of EVs, it often remains subjective to quantify the diameter of the smallest detectable EVs. To quantify the sensitivity of scatter detectors, we developed a new method based on photon statistics with co-funding from ISTH Scientific and Standardization Committee on Vascular Biology. Here you can see the light scattering histograms of a mixture of polystyrene beads, and in red our predicted resolution limit, which separates the bead signals from the background noise for different hardware configurations. In the end, this became such a technical story that with 22 equations, we decided to submit the work to Cytometry Part A. Naturally, once the referees have reviewed our approach, we will directly submit a layman summary to JTH. And please, let me know if you see any mistakes. The development of tailor-made reference particles or procedures to quantify the sensitivity of flow cytometers is not particularly something which will improve flow cytometry detection of EVs on the short term. So what can you do as platelet-derived EV researcher, besides starting to calibrate? So to achieve data comparison between clinical research studies, we should start reporting all details that are required to interpret the flow cytometry experiment. To ensure reporting sufficient details, the EV flow cytometry working group wrote a framework for standardized reporting of EV flow cytometry experiments called MyFlowSite EV. I can highly recommend to fill out the MyFlowSite EV template and add it as a supplement to your manuscripts. As an example, the first MyFlowSite EV compliant clinical research study was published this year in JTH. With calibrations and standardized reporting in place, time is ready to establish the first reference values for EV concentrations in human blood plasma. Therefore, we included 224 healthy volunteers and performed sample handling via the recent guidelines of circulation research. We applied pre-storage quality controls, like residual platelet concentration and serum index measurements, and we will measure EV concentration post-storage in a fully calibrated, fully automated fashion. Results are expected in the course of this year. Last but not least, I would like to draw your attention to the activities of the EV flow cytometry working group, which consists of members from the ISEF, ISEC, and the ISTH communities. I already told you about the minimum information that we recommend to report when doing a flow cytometry experiment on EVs. In addition, we are currently writing an educational compendium about EV flow cytometry. Finally, I can highly recommend to follow our bi-weekly EV flow cytometry seminars, which involve both educational and research talks. Thereby, I would like to acknowledge all my colleagues and funding, and I would like to thank you for your attention.